So while we wait for people to show up, I'll just put this back up again. So this is Wildlife Biogal and
Anna Boegehold.
Yep. So our Twitter handles are at Wildlife Biogal and
at Anna_BOEG.
And if you want to be able to check out this broadcast later, go to wildlifebiogal.com and you'll be able to see everything on either YouTube, get the Periscope link. I'll also put a broadcast out on SoundCloud later. So here's all the information if you want to be able to take a screen grab of that. And with that, we're back here at Wayne State University with our lovely PhD student here, and we're going to harass her for a whole bunch of information about her life as a PhD student, a little bit more about her research, things with community outreach, what it's like to be part of grad student union, how their union is here. So I think we're gonna like make this more of a discussion based. So anytime that you guys have any questions or anything, feel free to just throw them out there, and we'll answer them as they come along. So so why don't you tell us where you did your undergrad at, what brought you to Wayne State?
Alright, this is kind of a long story, but I started at Michigan State University, and I was there from 2006 to 2010, and I got my bachelor's in fisheries and wildlife with a minor in Spanish. And people always asked if I was going to go talk to Spanish fish when I told them what my major were. Won't believe how many people asked me that. It was awful. And then I took some time off. I lived in Montana for a few years. And then I lived with my brother for a few months in San Francisco. And at the time when I was living with my brother, I was looking into grad schools, and I looked at a lot of different places across the country, and I was trying to find something that I think would be a good fit. And then I was like, hey, I'll look at Wayne State in Detroit, because that's like kind of by where my family lives. Mm-hmm. And I found Donna Cashian, who's my advisor, and her, I don't know, just what her lab did, and like her, career progress was something that I was really interested in. She did a lot of aquatic toxicology work, which when I was at Michigan State, I worked in a lot of toxicology labs, so that was something I was interested in. And it just seemed like a really good fit, so I talked to her. I visited her when I, came back home to visit, and applied to grad school, and she made sure I got in. So yeah, that's kind of how I got here.
Yeah, that usually seems to be how it works in most situations, with, at least in biology or conservation biology especially, that it's kind of like more who you know, and then they get you in. It's not necessarily, I mean, there are still some people where they just straight up blind apply to a university, but it always seems like that advisor ahead of time, and they're the ones that like make sure that everything gets taken care of on their end with the university.
Yeah, that's definitely how it's worked in our lab with other students as well, and that's how, my sense of how it works in ecology in general. But here at Wayne State, it's kind of weird because our biology department is very heavily geared towards microbiology, and Wayne State's a huge medical research campus, so we have a lot of that going on, and it's the complete opposite for everyone else. Like in this building, like they think we're weird. Yeah, they blind apply and do rotations through labs.
Okay.
So yeah.
Yeah, I like the aspect of knowing your advisor and knowing that lab before you go in. I think it makes you feel a bit more secure in your decision to go to that place, rather than I don't know anyone and I just hope it works out. Here, like, you at least have, I mean, there still can be surprises and stuff once you show up in that lab. Like, maybe they weren't completely honest with you when they're, talking to you and stuff, but you like to think you get a good sense of who that person is, and that you're going to be able to get along with them and work them for, like, with your master's for the next 2 to 3 years, for, your PhD the next, possibly 4 to 5 years.
Yeah, I originally just wanted to do my master's, and then Donna convinced me to do my PhD instead. But, another good thing that I was able to do before I even applied was I went out to lunch with just Donna's students at the time. So then I got to, get some insights into what it was like in her lab without her there. So, you get the good dirt on what's going on in the lab.
Yeah, and they will usually be pretty honest with you about stuff and let you know what's going on. And
Yeah.
so yeah, I've had to do a number of those lunches of where, not necessarily with my lab, but it always seemed like other professors in our department were like, "Hey Nicole, you've been here for a little bit. You want to come out and tell everyone like what's going on with the university and stuff, and give them the rundown." And it really does. It makes you feel a lot better because I know where I'm going to school later. Like I've known the advisor for so long, but I've also known her students for just about as long, and I've been friends with them and been able to chat back and forth. And never hearing one bad thing about the advisor or the university or their department through any of them, through all my engagements with them, has really like helped me feel like very secure in my decision of where I'm going.
Yeah, especially if you're moving somewhere unfamiliar where you don't already know people.
Right.
Yeah.
I mean, they're basically your little lifeline to that community until you can start establishing your own ties around it, and you're very dependent on those people that they're basically your family. I mean, you're working with them all the time. So after Michigan State and then now here, tell us a little bit more about your research, for all the people who aren't listening earlier.
I came in at a time when Donna had got a grant with the USGS to work on a project involving dry-scented mussel reproduction. So I look at quagga mussels, which, if people aren't familiar, they're the sister species to the zebra mussels that inhabited the Great Lakes.
So those big bad invasives that we hear all about.
Yes.
Okay.
Highly invasive species. The quagga mussel is actually kind of interesting because they found them like in Lake Michigan at depths of like 90 meters or more. So they can survive at really great depths that zebra mussels can't. They can survive and reproduce at colder temperatures than zebra mussels. So, in some ways they're maybe even a slightly bigger or at least different type of nuisance, just because they can get into those deeper reservoirs out west, especially, is where people are watching out for them. So we're looking to see how cyanobacteria can impact their reproduction. If you've heard about like the harmful algal blooms out in Lake Erie or any of the Great Lakes, that's all cyanobacteria that causes that, and they're harmful because they can produce a variety of different toxins that can impact human health and wildlife health also. So I've been looking at how cyanobacteria can impact their spawning and their fertilization, and we've also looked at how they can impact sperm movement, and right now I'm looking at how lethal is cyanobacteria to quagga mussel larvae, which are called veligers. So I'm doing feeding assays with that.
The veligers, from what I kind of remember with the mussel presentations I've been exposed to up at Central Michigan University, those are the ones that when they get released, that they go up to the fish gills for a while and then they spread out that way, or is it? Is that the audits? Oh, okay,
yeah.
All right. So with the invasives, do they just go into the water column and then spread out themselves? They don't need that host to be able to distribute, or
right. So native mussels in the family Unionidae, their larvae need a fish host. They live in their gills to develop, but invasive dry-scented mussels, which are the zebras and quaggas, they're broadcast spawners, so males and females will release their sperm and eggs into the water column. At the same time, they'll mix, fertilize, and form free-swimming planktonic larvae called veligers, and then they're just on their own. They live in their veliger stage for a couple weeks, and then they'll metamorphose or metamorphosize. I think it's metamorphose, but that sounds weird. Anyway.
I'll call you the expert on that and not even touch it. So.
Yeah. They transition into adults, and they'll settle where they become the benthic, not sessile adults.
Okay.
Yeah, so that's the life cycle of the Quagga Mussel, so we're thinking that there might be some type of interaction going on between the phytoplankton community, since they are broadcast spawners and their young rely on, they feed on plankton, they're free living. So the theory was that mussels might be induced to spawn during times of year when there's a lot of diatoms, which contain a lot of fatty acids, green algae, which are better, and that so on the flip side, cyanobacteria might be harmful to the development of the larvae, and that might inhibit the mussels from spawning.
Okay.
And there's been some work done in marine bivalves and other marine invertebrates that show that certain algae can induce them to spawn. So we wanted to take this to the Great Lakes and see, if there's any connection between algae and invasive mussel spawning.
Okay. So with the research, how much longer do you have? What do you think you're gonna? That's always the question everyone asks.
Yeah. I think I've already been asked this today, twice. So I started in the fall of 2013, and I'm hoping to graduate in 2018. So that'll put me on a 5-year track, which I feel like is pretty normal these days, especially I just went straight to PhD. I didn't get a master's, so yeah, because Donna convinced me not to.
I mean, if you could still get all the knowledge and get the experience out of it, I see benefits, pros and cons to going straight to PhD versus doing your master's. I mean, it all depends, I think, on what you make of it.
Yeah,
that's the big thing. So, where are you going to be presenting this research at? What conferences do you normally go to for things like this, where people can hear a little bit more detail of research?
So we normally go to the Society for Freshwater Science conference, and I presented this research in May in Sacramento there, and I'll also be at the Upper Midwest Invasive Species Conference this October in La Crosse, Wisconsin.
Okay.
Presenting this, and then next year, IAGLR, the International Association for Great Lakes Research, will be here in Detroit in 2017. And then the Society for Freshwater Science will be here in Detroit in 2018.
Okay.
So probably those.
So you have plenty of opportunity to hear all of this awesome research. You did say something earlier that you have that Spanish minor, and I find that really interesting. How much do you think that's helped you with communicating your research and your science with other people?
Well, it definitely helps me learn the Latin names because it really does, because when you're learning new Latin names, if you know what the root word is, sometimes they're descriptive.
Right.
So if you know so Spanish is a Latin language, obviously, so I can figure out some of the Latin, and it helps me remember what the scientific names are, I guess. But also last summer, I had the opportunity to go teach a workshop in Puerto Rico.
Oh, nice.
Which was cool. Donna's one of her lab mates from her PhD at Wisconsin-Madison is a professor down there at University of Puerto Rico in Mayaguez. And he has a grant to bring students down, and the goal is to teach underrepresented minority students about STEM fields.
Okay.
So we went down and did a 2-day teaching workshop, and we were part of the grant was we had to stay to be exposed to the culture, so I was forced to stay in Puerto Rico for 2 weeks and eat the food. It was terrible. So knowing Spanish obviously helped me there.
Okay.
But going forward, what I want to do with, knowing another language is I'm hoping that when I get to do my postdoc, I can do research in a Spanish-speaking country.
Okay.
And also, I was telling you earlier that one of my major lifetime goals in science is to help increase the diversity in the field of ecology. I don't know, anyone who's familiar with the field knows it's not very diverse right now. So I just want to help foster an environment where more people feel welcome. Like if I can speak someone's native language, maybe they're less scared to, you know, go to a conference and interact with people, or yeah, because it's really hard to break into a field where no one looks like you or no one speaks the same you know native language as you. So yeah, I just hope I can help other people who might be, you know, scared to go to conference.
Right. Yeah. I actually, we got a question from Bobber4143. Why aren't there more lab science scopes? I don't know. We're going to try to do more of them. I hope we get a few more in. If you guys like it, definitely make sure you share this and let people know, and we'll be encouraged. Good. Yeah, we'll definitely be encouraged to keep doing this. I like being able to broadcast from where people work. Also hoping in the future that we can do some periscopes where people are actually out in the field, so you actually get to see some of the work that labs like these here at Wayne State, you know, other folks are doing, you know, where you can actually go out and see them sampling the water, see them, you know, pulling those fish out, you know, IDing them, so you get a really awesome sense of what people are doing. So that's also some hope. But yeah, just these, you know, sit down discussion ones, just actually where people are, it gives you kind of a sense. I don't know how well you guys can pick up on all the noise of all the machinery in here in the lab. Oh, we have another question from Bobber414. I have a home microscope. I'm the only microscoper on Periscope. So wondering, where are all the scientists? Yes, well,
I do a lot of microscope work here because when I do my spawning assays, like in order to see the Quagga Mussel sperm, I have to look at it under the microscope, and I do fertilization tests where I combine, you know, the sperm and the eggs to see if they'll fertilize in different solutions. So I look at, I'm under the scope a lot. So the belligers are microscopic.
We should get you to hook up your cell phone to your microscope, and then you could broadcast it out and show people. Because I think I've seen, oh, you have to show, yes, that's what Bobber's 4143 is saying. Yeah, and I think I've caught a few of your broadcasts, Bobber, and it's really interesting to be able to see what's under that scope and have it described, and the number of followers you get. That it's interesting, like, you know, even though like I've spent time under his scope, Bobber's saying I do art and science with music. Yes, he does have a very good music playlist that he plays. It's fun, so definitely.
I try and take like pictures and some video under the like microscope with my camera sometimes. Yeah. Like I posted a picture not too long ago of drycinid larvae under cross-polarized light, because when you put them under cross-polarized light, they glow, and there's like a little X in the middle of them. It's pretty cool. So, and that's how like I can tell them when I take a plankton sample. That's how I pick them out.
Yeah, like I've always told my students when I taught microbiology to bring their cell phones, and I taught them how to take the pictures like through the scope, so that way they could have them to go back home and study with. But what's even better would be to like actually see you live going through and picking stuff out and being able to teach people what it is you're seeing and all that. Like I think that's a really good idea, and something that I'm going to harass you to do until you actually do it.
So we have a microscope. It's more of a dissecting scope. It doesn't. It's on the other side of the lab.
All right.
That's actually hooked up to a camera that hooks up to the computer, which we use to take pictures of, mostly when we're IDing bugs, macroinverts, take pictures of Daphnia, stuff that's small but not super small. Stuff that you can see with your naked eye but want more detail of. But I also look at the belligers under that scope, so I can take cool pictures. And then also we do some sperm motility work at a lab in the pharmacy school at Wayne State, and they have a video camera hooked up to a microscope that goes up to 400x.
Okay.
So we're able to take video of
Yeah, so Bobber's saying if you scope it, I'll share it if I can catch it. So yes, that would be great for sure.
Yeah, it's really cool because we can take video of, you know, things at 400x.
Yeah. Well, and what would be nice with that too is you actually have a real Great Lakes scientist showing you this stuff, and I think you're going to be able to learn so much because you'll be able to like really, you know, they'll be able to ask you questions. You will know what it is you're talking about and be able to explain it. And you'll have fun with it too, because I always find sharing science makes it so much more fun with me. Like, you know, whether it's sharing it with my 6-year-old niece and nephew, or we will get her a Periscope. So Bobber's asking, does Anna have a Periscope account? We will get her hooked up with one after this. And she's on Twitter, so what's your Twitter handle?
ANNA_BOEG.
Yeah, there you go. So, we will get her to start periscoping, and
this is actually...
so see, you can just see he just took the screenshot with the little camera thing that popped up. Yeah.
This is my first Periscope experience.
Yeah. So we're going to make it a winning Periscope experience and make her have to stay on for many, many times to be able to keep doing this, because I think if we can get more and more scientists to be on Periscope and to be on Twitter and to share their science, not only is it good for us fellow scientists to be able to learn from each other, but it's a great way to communicate with the public and be able to share what it is we're doing. I think it'll help take away some of that mystery of what we do, because a lot of times people out there, they only hear about what it is we do once the news reports some little snippet of some publication that we submitted out.
Or misreports it.
Or misreports it, for sure. And this way, we have that direct contact with the public, and we can help alleviate some of those fears that they have with our science, and can really explain it to them in a way that the news might not necessarily be able to pick up and be able to properly translate the science to the general public. And I think we can do a better job of that. Bobber's saying, seriously, a lot of people on Scope believe in flat earth and moon conspiracy. Science education is desperately needed.
Oh my god.
Yes, I agree. Even just like things that aren't so far off, like you were talking earlier, we did an earlier broadcast with the advisor, Donna, for the lab here, and you were saying how you've had a number of people ask you, does the Army Corps of Engineers control the Great Lakes levels? And to me, that seems so far off. Like, why would they control that? How would they control that? But I'm a Great Lakes scientist, so I know how the water comes into the Great Lakes. But for a lot of people who don't deal with the Great Lakes, or maybe just see it occasionally when they go out to the beach, they might not know. And so right there is a simple misconception that some people have, that by talking to an actual scientist, they can find out that answer and be able to have that, myth dispelled, and hopefully then we'll be able to help educate other people that they know.
Our lab does a lot of personal interactions with the community too. We do a lot of community outreach. One of the projects that Donna is a part of is a fish consumption advisory on the Detroit River. So we make signs and put them up at popular fishing spots that tell you which fish are good to catch and eat, and we pass out pamphlets and actually talk to people and survey them about what they're eating. Are they cutting off excess fat on the fish? And just, advising them about different contaminants in the fish that could impact their health. So we do a lot of, traditional one-on-one style stuff.
Right.
We also, everyone in my lab volunteers a lot. We do community outreach. We do field trips with middle school and elementary school students and high school students. Sometimes they'll come to Wayne State and we'll set up a lab for them. I've gone to elementary schools to do guest lectures on invasive species.
Yeah, we started, I think 3 or 4 years ago, we started what we call BioBlitz program.
Oh yeah.
So I don't know how many people out there have heard of Jane Goodall's Roots and Shoots program, but she came to CMU about 4 years ago, gave a talk, and during that talk she talked about her Roots and Shoots program, and so we really liked that idea. So we took that idea, formed basically a very similar thing of where we go into elementary schools and we educate kids about science. We don't just do biology, we branched it out to physics and a few other things like that, but I think it's so important to hit the kids at that age to get them excited about science, so that way they grow up with an appreciation of science. With my niece, who's 6.5 years old, I got her a bird book, and now she's all excited about going out and learning birds and things like that. And so she's carrying around that book and having so much fun, with her binoculars, trying to see what birds are out there. And it's all because she was exposed to that. And so by doing that outreach and really hitting those kids, I think it's important. That's the thing I like with you guys, is you're actually bringing them into the lab. That's one thing I wish we could start doing at CMU, is actually start bringing those kids into the labs, and then even doing some field trips, so with taking them out and letting them see the science and be part of it. I think that would really help cement it in, not only a love for science, but just a love for your environment and respect for it, and help, foster that, because so many people don't have that.
Yeah.
Oh, we got a question here from Bobber. I think adult science is important too. A lot of ignorance, even by high school science standards on here. Yes, I agree with that too. Yeah, I mean, for sure. I mean, you've got to hit the whole spectrum. But I also find too, if you hit kids, they also go home and start sharing with their parents and stuff too. But you could also say the opposite too. If you teach parents, then hopefully that they would share with their kids as they're growing. So it is important to, make sure that you're still hitting that full spectrum of your outreach program.
We also participate in a lot of citizen science, where the community goes out and volunteers. We have one of the local watersheds, the Rouge River watershed, where Henry Ford had his infamous Rouge plant. So that's historically impacted, like, watershed, especially as you get like into industrial areas. So we have a group called the Friends of the Rouge, and they do water sampling twice a year. They do, and that's a community thing where everyone goes to 2 different sites and you look at what insects are living in the water, and that can be a proxy for stream health. And that's really fun. I see a lot of people from all age groups do that. You have adults that are really into it, and then you also have kids. Some people take their Cub Scout groups to do that. My cousin's about to turn 14 this fall, and she goes with me. She's been going with me for the past few years. So that's really nice.
Yeah, no, I like the idea of hitting, the Boy Scouts and Girl Scouts, like those, because they're always looking for those outdoor activities to do, and I think you've already got like that solid group that you can just pull on and say, hey, come hang out with us, we'll teach you something, and you get that experience.
Even adults, like saying, hey, come out and help us, like, we need you, like, if you make people feel useful and like, make science like not something that you do in a secretive underground lab, but like something that they can do too.
We don't always just sit around going moo ha ha ha.
Yeah, if you show people that, like, hey, you can help with this, like, you can actually come out and do stream ecology with at the same level that professionals do. I think that's fun for people too.
Yeah, I think that's a really good point. Is while they might be doing some simple stuff, it's still quality science, and you can use the data that they collect to actually make some really cool insights into what's happening out in the world. And I think when you make them feel like, hey, you're part of this really big thing that we're doing in this lab, this is real true science that you're doing, I think it really truly encourages them, because they feel cool. Like, it feels cool to do that.
Yeah.
So I guess what other things you do? A lot of outreach. I'm going to try to steal these cards. I've got my mother tagging along with me on this big, huge SICOM adventure, because we're also going to a wedding in the middle of it. And ever since Anna handed off these cards to her before the broadcast, my mom's been playing with these cards. So I think we can say that they are a quality product here. Yes, we're going to steal the cards from you that you've been playing with for a while. So I don't know if you want to explain these cards.
Yeah.
So we've got these awesome cards. I don't know if you want to, like, pull on. Oh, you gotta do the crayfish.
Okay.
I've got a good friend of mine who's the wedding I'm actually going to this week is a crayfish research scientist. He's starting his PhD at Ohio State.
Cool.
Maya Algalon. So if you like crayfish, talk to the crayfish man at Ohio State. But
we made these cards. I actually didn't. Other people in my lab did. I can't take the credit, but I still think they're really cool. They're Great Lakes Aquatic Invasive Species watch list cards. So this is the front of each card, basically they're watch list species. They're species that aren't in the Great Lakes yet, but we're concerned that they're going to show up any second. So we hand them out to people who are going to be on the water, boaters, community, like lake association groups, so people get familiar with them. And each card is a different species. So this one has a crayfish on here that might pop up in the Great Lakes, and then if you see at the bottom, it gives you like a little blurb about them. And this one just says the species may threaten native crayfish. Oh my gosh, this is a funny Latin name, Cherax destructor. So yeah.
Oh, and I like how you guys called it the yabby too. That's what were the Australian way of saying crayfish, if I remember right.
Oh,
I'm
really?
trying to remember, Mile. He taught me once. We also had an Australian scientist come in, and she was all excited when they started talking about yabbies.
Maybe
So.
that one's from Australia?
Could
I'm
be.
not too
I don't
familiar
know.
with crayfish.
We'll have to hassle Mile and have him give us an answer on. Yeah. So yeah, definitely. Yeah. Yeah, because you got your carp in here.
Yeah, we have a lot of different things in here.
Yeah, but there's some in here like I've never even heard of. So this is like I'm learning as I'm flipping through here.
There's fish, like bivalves. Here's an invasive daphnia, so that's zooplankton.
Yeah.
So it's a whole range of
here's
stuff.
your mussels here. Oh.
Is that the golden mussel?
Yes. Yeah.
Some plants. So.
Yeah, and then like you guys have this, information along with like the playing cards. There's also this little write-up about what are invasive species, how they're spread. So there's also some quality information in here along with something that people can play with.
If you message me a shipping address, I might be able to swing it.
Okay, so the question was, how do we get them? So yes, send the messages over this way, and we'll see what we can do.
I'll see what
I've got mine here, and I'm not letting them go. So.
Yeah, I'll see how many extras we have.
Yeah, because these are really cool. This is a great visual way and a fun way to get people engaged and learning, and like you're playing with these and you're just gonna learn about them as you do it. So
I mean, the idea is for them to be like flashcards. You're playing with them over and over again, so you're seeing them. And it gets ingrained into your head.
Yeah.
Next time you go fishing, you find a Asian carp or something.
You're like, oh boy! But that way you're teaching them about invasive species. They have a healthy respect, and they're also help to be like those second set of eyes that the scientists can't have, because we can't be out there all the time monitoring. So if you can have citizens out there monitoring and they know what it is that they're looking for, you can get that early detection so much faster than what you would with just your random net trolls and things like that we do. So
and that's fun. It's not always fun going and telling people that the fish that they're eating is filled with contaminants and they shouldn't eat it.
Yeah.
Because they don't like that, and they don't like you telling them to stop eating their favorite fish that they've been eating for 50 years.
You want the cards back to play with? Yeah, she does. It's quality stuff when the cards are more interesting than Facebook. So. Bobber says, I get asked how clean the water I scope is all the time. So so you guys do some water analysis. Like, so how is the water around here that you guys deal with?
It varies. So, we're in Detroit, historically very industrial city, very industrial area. Detroit is actually like the epicenter. People are really sensitive to food and water cleanliness.
Yes.
Yeah. We're actually like at the epicenter of a lot of the invasive species. Like the zebra and quagga mussels started here in Detroit because we're a big shipping center. Emerald ash borer was started, came into the Detroit area through just like, shipping of wood and other things. So yeah, I mean, there are a lot of issues here, and I don't know what you mean by clean, because we've also had, so we have a lot of invasive species, obviously, but then with the industrial side, we've also had a lot of contaminations. There's like a PCB leak, actually, in the southern part of Lake St. Clair. So if you're not familiar with the Great Lakes, Lake Huron kind of drains into the St. Mary's River, which goes into Lake St. Clair, which drains into the Detroit River, which goes into Lake Erie. So there's a PCB. People freak out when they see, oh my god. People freak out when they see protists and want to know where they can avoid them. I don't know, protists contaminate like everything I do, so I don't think you can avoid protists. They're a lot scarier. People hate the idea of gut flora inside them too. Yeah, there's a lot scarier things, like that PCB contamination site. Like I'm way more scared of that than I am of protists. But yeah, just from all the car plants, we have an oil refinery, on the Detroit River too. So there's a lot of more scary things. But one of the cool things that we're doing at Wayne State, so if you missed the earlier Periscope, we collaborate a lot with different departments.
Just give everyone a visual of the lake system that we're talking about here. So just showing how the St. Mary's, Lake St. Clair, and Detroit River, and then the Great Lakes all relate there. So
yeah,
this right here in the center, that's the mitten of Michigan. So,
but one of the cool things that we're doing is, so we collaborate with people in the pharmacy school, the medical school, and then civil and industrial engineering, and we have a couple new like field stations, and one of the field stations that we have is actually a drinking water processing plant, I think, that's right on the Detroit River, and inside of the processing plant we have a smaller replica model of the same process that the water goes through to be clean for drinking water. And so what we can do is do research on contaminants there and see what contaminants are in the Great Lakes can affect like zooplankton and fish, and also see what methods work best for cleaning those contaminants, that, you know, the chemical contaminants out of the water. That's cool.
Yeah. So you've also done, because I was, you know, stalking you online before I showed up, but I saw you also do some Phragmites research that you've done.
Oh, yeah, that was, so one thing that's great about Donna is that she's like always she's always a champion for her students. She's always making sure we're funded, making sure we can go to conferences. So the thing I did with Phragmites was, the summer before I started my PhD, she got me working in the lab before I actually started, so I could get going right away and have some funding the summer before I actually came in. So, and that was actually how I got to go to my first conference, was through that grant. So it's not really related to what I do, but
so that was just like some initial work that you got to do.
Yeah, it was a way for me to get to a conference, get to Portland. So
yeah, see, for me, like, since I do mute swans, I'm like really interested in invasive species. So anytime I see something about an invasive species, I'm like, oh, okay, what'd you do? What'd you find out? What's going on?
Yeah.
So yeah, Bobber was making a good point with Periscope. Is, yeah, you do have a lot of people coming in and out during your Periscope talk, so you do have a lot of the same questions asked over and over again as you're doing stuff. So yeah, there are times where you're doing a broadcast that you can be repeating yourself as you have people come in and out. So it's nice in that format because you can have people come in and out at their leisure to watch stuff, but it also can make the broadcast kind of repetitive sometimes as you have people asking the same stuff, and you could say, so what was your research again for the 5th time? And so sometimes it's nice to be able to, if you do shorter broadcasts, and you can keep that repetitiveness from happening, and then you just force people to have to watch it later. But then also, if you extend the broadcast out, then you give people time to get in and then actually have that like engagement live, because that's the whole point of Periscope, is you can actually have those people that you can talk to live, you know, rather than just YouTube. Because like if we wanted to just sit here and do an interview, Bob was saying you can learn how to be succinct but informative. Yes, exactly. So like if we wanted to just straight up do an interview and put it out there for everyone to see, we would just do a video and then put it out on YouTube. But the nice thing with Periscope is you do get that interaction with people live, and I think it leads itself to more interesting discussions, because you'll have people in the audience that bring up really interesting points, and it gets you thinking about your research or other topics in ways that maybe you hadn't, and it can make for a very lively discussion. One of the very first Periscopes I did was at Agler with Katie O'Reilly, which, if I remember her right, her handle name is Dr. Catfish. And she's out. It's Bob was saying it's a hangout atmosphere almost. Yes, and it definitely can be. That's one of the benefits with it, and that's what it felt like when we did our first broadcast. It was just the two of us talking, and we had everyone coming in asking questions, and it made it such a fun discussion. And we were just talking about SICOM, and it made interesting. So Bobber's saying, like, how we're hanging out now. Yeah. So, I mean, Bobber, I don't know where you're at, but wherever in the world you're at, you're getting to, like, hang out with us. I mean, we're sitting here in the middle of Detroit talking about science and Great Lakes, and you're getting to be a part of it and have that discussion with us. San Francisco Bay Area. So awesome.
My brother's flying in from San Francisco on Friday to visit. Yeah, that's
so you should just hop a flight over and just come on over and come hang out in the lab here.
Yeah.
So,
I try and go there at least like once a year.
Once a year. Yeah. Well, then you can go hang out with Bobber.
Yeah, do some science while I'm there.
Yeah, you pick up some tips and tricks on how to broadcast your microscope work on Periscope.
Yes,
and yeah, there you go. See, you got your first Periscope tweet up all planned right there. But no, it really does help with that interaction. You can get these, things where you meet people. You have I always forget how you say that. The tardigrades. Yeah, I know the water bears.
Yeah.
Yes. Yeah, I think I actually caught part of that broadcast, Bobber, of where you were, we had the water bear out, because I'm not going to try to say it, because I know I'm not going to say it right. But it was really cool, and so, like, it was just poking around and showing it and talking about it, and it was so interesting. And I think with being able to show off all of those Great Lakes species that you have around here, and there's so much interesting stuff that you would get people in. Bobber gets, I think you said once that you had like thousands of people watching or something. I'm trying to think of how many people I've seen most, like, on some of your broadcasts. You always seem to have like hundreds of people commenting and things like that, and it's just like it's a non-stop stream of questions and things that he has going on there. It's really interactive, and it's so much fun to, like, just watch. Bobber says, the water bear scope had 24,000, I think. Yeah.
People love water bears.
Yeah. It's cool stuff. And, like,
I have one of those, like those plush. Have you seen those plush microbes?
Oh, yeah.
I have a water bear that I was the maid of honor. That's the best lady, right?
Yeah.
At my friend's wedding last year, and she got me a bunch of those, like, plush microbes.
Ooh, I have to be in a wedding for one of our former grad students that she does Great Lakes stuff. I'm gonna have to see if I can get some, like, fish or something along those lines. Barbara said I did paint drying the other day. Lots because of the novelty. Yeah, so you can have a lot of fun with it. So, you don't even have to just do your species, but you could do, like, other things around, or, like, bring other people in. You could have so much fun with that, and I think that'd be such a great way to do outreach and stuff, and you could hit so many people, not just in the Detroit area, but all over, to teach them about what you're doing and really educate them. That would be really cool.
Yeah.
You're gonna have to think about that.
Yeah.
You would have fun with it. And the nice thing is, like, because, like, when Bobber does it, every time I've seen you just have the camera just on the microscope. Like, it's never off of that. So it's just, your voice going on with some music in the background, and the only visual you see is what's underneath the scope. And so I think it's, like, a very comfortable environment. Like, you could just be sitting there in your PJs, as far as we know, broadcasting. And
I always wear my pajamas to the lab, so that would work out.
I'm usually, like, in my workout gear when I go to lab, like, unless I'm having to teach that day. But so it's, like, just a very comfortable environment where you're getting to teach people. And then what would be kind of cool too is, Bobber says, I put the smartphone out to show the actual sample sometimes. Oh, okay. Yeah. So you just, like, pop it off there, and then, like, show the stage there with everything, so you can kind of see it more in, like real size of what it is. Yeah, that's a good idea. Yeah, so that way people kind of get a sense of what it is they're actually looking at, not just, like, the zoomed up thing. So yeah, I can't wait to see your first broadcast. I'm already, like...
So you're already committing me to...
I'm committing you to doing this.
I haven't even agreed to it.
Yes. Well, you guys have all the tools here to do it, and I think it would be really cool.
We have, like, 1 million microscopes.
Yeah. So, I'll have to see if I can get, some other... Because there's definitely a lot of folks at Central Michigan University that do a lot of microscope work with the species they do. Yes, Bobber saying you have no excuse. I like that. High five to you for that one. With all the macro inverts that they work with and stuff, to be able to have those under the scope, like, just teach people how to identify them and things like that. Because, how do you tell? Oh, thank you. High five back. Yes. what did we do before emojis?
I don't know. Yeah, I don't know.
But yeah, to be able to like learn how to identify, like, what's the difference between a mayfly and a stonefly and a dragonfly? You
Okay, I know
larvae
that,
and stuff.
but like when you get down to like, you know, family and
Oh,
genus,
yeah,
like,
yeah.
I have other lab mates that can help with that part.
But I mean, even just like the journal, like, you know, being able to pop out that lower jaw of a dragonfly would be so cool for people to see. Like, that's the thing that always I find is the most entertaining.
Kids,
yeah, like, I mean, I took my niece out to, I forget what lake it was they were at that was over by their hometown. And so I was just pulling stuff out of the water and got one of the dragonfly larvae and I popped it out and she was just so mesmerized with it. So at 6 years old, I've got her hooked on, you know, macroinverts and Great Lakes science. Yeah, and you know, you have to visually show, like, you can't describe that. You have to like show them how that like jaw pops out, and it's just it makes it so cool. And if like you could do that, I mean, like you could set up like things where you could broadcast to like the elementary schools and do that. That would be really cool.
Yeah, so multiple schools or classrooms could be involved in that at the same time.
Yeah, exactly. Especially like maybe with ones that they might not have the funding to be able to get all the kids in here, but they could like set it up for like an hour at their school to be able to like tune into this.
Yeah, I do local school visits.
Yeah,
I have some teacher friends, so like, I've gone out to elementary schools.
Mm-hmm. Yeah, but that would be like a really fun way. And Periscope is free, and all you need is someone with, you know, a laptop, and they can broadcast it right out. Like most elementary schools that I go into now, they've got the big whiteboards where they like, you know, they can do and so they can broadcast everything up on there for the kids to be able to see. Yeah, so I think that's a really good idea. I like that a lot. I'm totally committing you to so many things right now that you don't even know. You probably should just shut me up before I commit you to. But no, I think, that would be really interesting. So I like that idea. So
that would be a good way to get, because last year I coached a science Olympiad team at one of the middle schools around here. And like, it was kind of a far drive from my house. So that would be a cool, bless you. Sorry. It was probably right into the microphone. That would be a cool way to like teach, you know, like teach my science Olympiad kids if they couldn't make it to practice or I couldn't make it to practice or something.
Yeah. Cool. That's great. Well, I don't know, you want to talk about how your union is here for a little bit and then we'll let you off of here? Because I think we've been talking for quite a while already. Yeah, we're almost about an hour. Yeah, so we'll just do a couple moments about your union and then cut out and let us get some free time away from the camera.
Sounds good. So the other thing that I do that I think is really important, especially for grad students, is I'm vice president of my union. And like I was saying on the other Periscope, I think it's really important because at Wayne State we have a really good funding package for grad students who teach. I think our wage is really competitive for what other Michigan universities have, and we also get health insurance, which is huge. I mean, I had my appendix out last summer, which like it would have been really expensive if I didn't have, you know, health insurance. And that's obviously something you can't like wait to do.
Right.
So yeah, and we have all of those benefits because our union is really strong and we negotiate for those things. And when the university wants to cut our pay or take away, you know, our health insurance or raise our parking rates, we're there and we represent the students and we remind them, you know, we get paid like 1% of the budget but do 30% of the teaching. So, you know, that's probably not fair. And we do have a lot of support. So if we do need to get petitions signed and take action, we have the force and momentum to do that just to make sure the grad students are protected. Another thing that we do is we try and stand up for our international students who they have to pay different fees than domestic students do just to keep up with all their paperwork and stuff. So we negotiated that they can get reimbursed for those fees through the university because that's an unfair burden that they have to pay that you know, other people don't.
Right.
You know, we are trying to implement affordable childcare for grad students.
That's a good idea.
On campus who have kids, because we do have a lot of people who, live in the area and commute in.
Mm-hmm. Yeah,
and
especially as you get to those upper reaches of you're going to have your PhD students are more likely to be married and have families, and those are going to be in need of that. And if you want them to be able to be successful in their program, you have to help take care of them.
And some people might look at the union as, we just want to get everything we can from the university.
Yes, we have $100,000 in our pockets every year. It's not that, but it's you want to be at least taken care of to be able to survive and get through. You don't want to have to be living on food stamps. I know at CMU we don't have health care there, and that's one of our big things that we would like to see happen is actually get health care, because not everyone falls under the protection of their parents' health care. I mean, even though that age has been raised, you have so many grad students who are past, 26? Who are past that age who actually have to thus take care of their own health care. And if you can't afford, say when your appendix comes out, sorry, you're going to quit your program to go get a job to be able to pay for that, or you're not going to be able to have that, health crisis taken care of, and it still is going to impact your ability as a grad student to function properly. And so I think it really helps the university if they really support their grad students, because you're going to get a better product out of it in the end. It might be a upfront cost, but the back end of it, the publications that they're getting, the notoriety they're getting, the fact that you have grad students saying, hey, this university is awesome, and it's taking care of us, it's going to bring in more grad students and thus more research to your university.
Yeah, so even though we're only here for 2 to hopefully not much more than 6 years at a time, we do a lot of the teaching, so we are the face of the university for a lot of the students in every department. This is true not only in science. We do a lot of research for the university. We should be compensated fairly, and not just surviving.
Mm-hmm. And we understand too. Most of us who come into grad school, we get our tuition waived, so there is that added bonus that we're getting in addition to the pay and stuff. But there's the simple things like healthcare, and you said, the childcare would definitely be a big benefit for a number of students. Mm-hmm. That really would make your time at a university that much better, and then the product you're producing is going to be that much greater, because if you're stressing out of trying to take care of your kid, or how do I pay to go to the doctor, it really does weigh on your mind.
And anyone else who's a graduate student, you don't just show up for a crappy paycheck. You're dedicated to that graduate program, and you're very dedicated to the work that you do. And we work a lot on our program, so just being able to ensure that people aren't being taken advantage of, especially international students who might not be aware of some of their legal rights and other things, or might be too scared to stand up for themselves because they don't want to lose this spot. I don't know, I just really enjoy it. Plus, again, tying it back into the whole SICOM thing, I really like getting to know fellow grad students throughout my university in different departments, because it gives me better insights on maybe how I talk to others, or it's just nice hearing what other people do, and then you might be able to take part of that back to your work.
Right. Well, and I think it helps too. I mean, the more grad students talk back and forth with each other, not just within their own department at their own school, but in various universities across their state or the U.S. or beyond that, I think it makes everyone better, because you see what some universities are doing. Oh, that worked, or that didn't work. How is our university doing it? Does that work for us?
How can we help Central organize to get healthcare?
Right. Yeah, and maybe there's things that we do that maybe you guys don't have that might be oh, we never thought about that. That's a good idea. Mm-hmm. Yeah. So, and having either through Periscope or social media, or just meeting up with people live and in person, it really does help. And what's nice now is after this, I've got someone that, having met them in person, I can talk to them, and I feel much more comfortable with having maybe those more in-depth discussions about things. And so it gives you that,
reaching out and asking for help when you need it.
Exactly. Yeah. So, but anything else you wanted to cover before we sign off, or
no?
You good? Okay.
Yeah, I hope to see everyone at IAGLR in 2017 and SFS in 2018. I don't know. I love Detroit and living here.
Yeah, come check out Wayne State.
So.
I mean, I've got to say, I've only been here for afternoon, but so far the experience has been really good, and you know, it's definitely worth a swing down. And if you guys come to Iagler, you'll get to see Wayne State live and in person, and
a lot of fun, you know, activities planned that you can only get in Detroit. So,
yeah. So with that, we're gonna sign off. I'll put up the Twitter handles for everyone again. So I'm Nicole Wood at Wildlife BioGal.
I'm Anna Boegehold at ANNA_BOEG.
And if you want to be able to check out this broadcast later, go to wildlifebiogal.com. I'll have everything up on YouTube. I'll put out the podcast in a little bit, so in case you can't watch it because you're driving along between field sites, you know, it gives you a way to be able to listen to it. Other than that, if you have any other questions, feel free to tweet us, and we'll try to answer them when we can, because yeah, we'd love to keep this conversation going, and SICOM definitely is, I think, a great way to be able to help scientists and the public be able to reach out to each other and communicate in a way that hasn't been happening before, and I think it's going to really help bridge the gaps that aren't there. So with that, we'll say see y'all later, and have a good one.